joining us online as well. Welcome everyone to our annual Richard E. Weitzman Lecture, but a very special one today, honoring our most recent investigator, honored with the Richard E. Weitzman Investigator Award, Dr. Lynda Polgreen. For background information, by the way, these are all being recorded and they are available on our virtual innovation library online. So you can also send friends there if they couldn't make it today. So I think everyone knows Lynda really well, but I'm still going to do my intro because I've gotten to know her this past year, and it's been one of the great treats of being here at the Lundquist, along with getting to know so many others of you. In my opinion, her work absolutely reflects the very best of TLI's commitment to translating discovery into better healthcare for everyone. She's a dedicated physician, scientist, and mentor whose research has advanced our understanding of the treatment of rare pediatric diseases, bringing new hope to patients and families around the world. And I think you're going to see a little bit about how that works today. I actually got the treat of visiting with her and her team, working with these patients one time, and it was phenomenal, a little bit moving, I have to say, to see how important that work is and how much it means to those families. You really can't overstate the value of what she's doing for the families that she's serving. And again, that work, I think, exemplifies the very best of what TLI has to offer. When we bring all cylinders firing together, it's incredible what can be done here. Real testament also to the importance of the research that our investigators are doing. A little bit of the standard background materials. She got her degree in medical school at Mount Sinai School of Medicine in New York City, which is fairly cold, and then decided maybe it wasn't cold enough. And she went and got her pediatric endocrinology fellowship at the University of Minnesota Medical Center in Minneapolis, and a pediatric residency there as well. She's board certified in pediatrics and pediatric endocrinology, and she was initially on the faculty. You hung out there in Minnesota for a while at the University of Minnesota Medical Center. But she finally decided Southern California is the place she ought to be, and she came here in 2014. And if anyone knows the rhythm of what I just said, you are just as old as me. She's a clinical director of the Center for Treatment of Rare Diseases and associate director of the Institute for Translational Genomics and Population Sciences here. She's a real leader amongst our faculty. Pediatric endocrinologist at Harbor, and she also does work at Children's Hospital of Orange County. And as the director of the Center for Treatment for Rare Diseases, she really leads a lot of groundbreaking discoveries. I think we're going to hear a little about that today, so I don't want to say too much there, but especially around rare childhood inherited diseases, included diseases like mucopolysaccharidoses, or MPS 3, which is far easier to say. Sanfilippo syndrome has a more pleasant rhythm on the tongue, but this is a really challenging disease, and you'll learn a little about that from her today. But in addition, she does work on osteopetrosis, which these conditions, there really aren't really good treatments for. And she's working hard to find treatments that affect either the symptoms or the disease. And so we need a lot of work done there. It's very urgently needed by those families. She's shaped global research in many different ways, broadly going from developing a better understanding of how the diseases progress through multi-site natural history studies that she's been doing, as well as testing out new therapies in investigator-led clinical trials. So the most recent paper that a very high-level profile was in Nature Medicine, where she was looking at a trial of anakinra for children with MPS 3. And that work is continuing to progress. Hope maybe we'll hear a little update today. Behind her research, she works closely with patient advocacy organizations and serves on the National MPS Society Scientific Advisory Board. She advises industry partners in clinical design and monitoring for rare diseases and therapeutics. And kind of wrapping it all up, she really is a truly compassionate pediatrician who's a committed mentor, one of our true leaders here at TLI that I've come to rely on, previous board member for the TLI board, and really exemplifying overall that sense of collaboration and innovation and heart that really defined the Lundquist Institute. I truly can't say enough about her, but I'm going to have to stop there. So it's my very special pleasure to introduce and to welcome her today for her Weitzman Lecture entitled 25 Years After Aldurazyme, The Expanding Landscape of MPS Treatment. Thanks, Lynn.
All right. Thank you, Dr. Ramos. I appreciate that. Kind of extensive going through some of the work I've done, because for this talk, I'm actually not going to focus a lot on what I've done, but talk a lot more about the history of work that's been done here at Lundquist and as well as the UCLA Westwood campus that really was part of the reason I ended up here and supported the work that I have been doing since I've been here. But before I get to that, just like to take a moment and honor Dr. Richard Weitzman. Dr. Weitzman was an endocrine faculty here at Harbor UCLA. I don't know if any of you knew him. He was before my time, but from what I understand, he was very influential in the field of hypertension, doing research on neurohypophyseal hormone and cardiovascular endocrine physiology. So just as an indication of his broad impact on the research that he had done, there's not only an award here in his honor, but the Endocrine Society, our main large endocrine society, has an award in his honor every year as well. Okay, so 25 years ago, hence the name of the title, almost to the day, Dr. Zaimokakis and Dr. Elizabeth Neufeld published this seminal paper on the first in-human clinical trial of enzyme replacement therapy in MPS 1. This paper showed that there were improvements in both the biochemical and functional outcomes in patients with MPS 1 and was the very first treatment to show any efficacy in any of these lysosomal storage disorders. But to tell the whole story, we really need to jump back 33 years earlier. And I love telling this story to people who are working in a lab, particularly students, because the way I heard this story was that this incredible discovery that has really led to the treatment of many patients with a variety of different storage diseases started on an accident. There was a student in the lab, and I'm sure some of you have heard this story, but I'm going to just tell it again. A few of you might not have. But as I understand, there was a student in Dr. Neufeld's lab who had accidentally mixed two different types of MPS cells. So one from MPS type 1, or Hurler syndrome, and cells from MPS 2 called Hunter syndrome. And when they went back to their experiment, the cells didn't have the characteristic storage in the lysosomes anymore, and they thought they'd made a mistake, and they were going to throw it out. But fortunately, at some point in there, somebody had the right idea that, well, let's figure out why. And so what they then did was they cultured the cells separately, and then they cultured the cells, as you can see here. I've been told this pointer works. Oops. Oh, that. Yeah, there you go. It's the bottom one. Oh, thank you. Okay. Got to get the angle right. I'm going to use this. Does this work too? Okay. So what they did, when they had the two cells in the one culture, this is what they initially noticed, that there was this lysosomal accumulation that was corrected. So they look at the cells separately, and then they put the cells in a system where the supernatant is shared, the media is shared between both cells, and they found that both of the cells were corrected. And from here, they figured out that in a lysosomal storage disease, that lysosomal enzyme is secreted and can cross-correct other cells if that enzyme is near. And just to explain this a little more in detail from the schematic here, they later went on to really define this whole mechanism by which this cellular cross-correction occurs with the cells from the healthy cell, which you can see here, for example, on the top being the enzyme sufficient cell. They will secrete their lysosomal enzyme, which can then be taken up from other cells that don't have that enzyme through the mannose-6-phosphate receptor. That enzyme is then trafficked to the lysosome and fixes the deficiency in that deficient cell and clears that storage material that accumulates in these diseases. So moving forward, I see some smiles in the audience. I don't know if this bungalow is still on campus, but a few years later, Dr. Kakis and Neufeld had determined how to produce the enzyme with the appropriate glycosylation in Chinese hamster ovary cells. and made the enzyme here in the C3 bungalow. And these photos are from Dr. Patty Dixon, which she was kind enough to give me years ago. But I just love them, especially the one of seeing the enzyme with the aluminum foil on the top here. This was not for human trials. This was for the dog trials that they did, because they did go on to show efficacy of this enzyme in a canine model of MPS 1, and then moved that forward into the clinical trials. And at this point, I'm going to pause talking and show this segment from 60 Minutes that picks up the story as it moves into human clinical trials. This way, you can learn what MPS does to the body from the patient and the parent, because I haven't done the usual intro slide of here's what MPS is. So I want you to, I think you can hear it, you can see it in this video clip much better than I would describe it in a slide. It also gives some insight into the experience of patients being in a clinical trial and the importance of patient advocacy groups in the field of rare disease research. Oh, sorry, I realized I didn't. There we go.
What can you do if someone you love is diagnosed with a disease so hopeless and so rare, drug companies won't develop a treatment because it would never turn a profit? You might try doing what Mark Dant did. His little boy Ryan was one of only 40 babies in the United States born each year with a fatal genetic condition called mucopolysaccharidosis, or MPS 1. Mark and his wife Jean were told they could do little more than wait for their boy to die, but the Dants were not willing to accept that. Ryan's father embarked on a mission with only one goal: saving Ryan.
When you hear the words the night before, what's it going to be like when I die? The next morning you go out and you say, you're not going to die. You're not going to die. And I'm a police officer, but I will find something, somewhere, somebody will give me the answer that will be able to tell you, Ryan, you know what? You're not going to die.
Hi.
Ryan was born in 1988, and from what his parents could see, was growing into a healthy, active toddler. Mark was on the police force, and Jean worked in the airline industry. Together, they were building a quiet, peaceful life in the suburbs of Dallas. That is, until 1991, when Ryan turned 3, and a physician discovered his liver was abnormally large.
What did he tell you?
Ryan was very ill, and that he probably wouldn't live beyond the age of 10.
He told you that?
Yes.
That day?
That he had a disease called MPS, or mucopolysaccharidosis. He said that Ryan would be probably blind or deaf or mentally handicapped, and he would be in a lot of pain because his limbs would draw up, his hands would change, and he wouldn't be the same boy.
Kids like Ryan with mucopolysaccharidosis 1 are missing an enzyme that the body needs to break down certain waste materials. Over time, the buildup damages organs like the heart, stiffens the joints, affects breathing, and stunts growth. In some cases, it can lead to severe brain damage and mental retardation.
You must have asked them, treatment, cure, what can you do for our son?
Yes, the answer was the same. There aren't that many children in the world with MPS, and because of that, there just is not much going on in the world as far as research.
If researchers wouldn't look for an answer, the Dants decided they would. While Jean stayed home with Ryan, Mark tried to learn just what he was up against. He went to a conference in Denver where he met other parents and MPS children.
Ryan was 3 when I left Dallas and went to Denver, and he wasn't that affected by MPS. But when I got to Denver, I saw 5 and 7 and 10 and 12-year-olds. But I didn't see anyone after 12.
Scary.
Yes. Children who were dying and children who by the next year would be dead.
And you couldn't see your son that way?
I would lay in bed at night and worry about Ryan being that way.
Mark became obsessed with finding a cure and started the Ryan Foundation to help fund one.
If everybody on the sheet comes in, we're at 141.
It became a community project with all their neighbors joining the fight. Their first fundraiser was a bake sale.
How much did you raise?
The day netted us $342. But it was a beginning. It's the first step.
$342.
With nowhere to spend it.
With nowhere to spend it, and how much did you think you needed?
I had no idea. I think what we needed was one day. Whatever money you could raise in one day, somewhere, somebody could use it.
Meanwhile, Ryan's health continued to decline. He began having headaches so excruciating he would vomit and pass out. His fingers started to curl. At age 7, Ryan's hands got too stiff to play his favorite sport, baseball. To keep him in the game, his father attached Velcro to his bat. Eventually, even that didn't work, and Ryan had to quit the team.
On Saturday mornings, when everybody else was going to their baseball games and soccer games, Ryan and Gene and I would go to a physical therapist. And on the way home, we wouldn't drive by the baseball fields. I would purposely take a long route.
So you wouldn't see the other children out playing a game that he loved but couldn't play?
And on a selfish perspective, so I wouldn't see it either.
Mark still worked full-time as both a cop and as Ryan's dad. Every day after walking the beat, he changed his uniform to walk another beat, knocking on doors and shaking hands,
Very nice to meet you.
raising money to give to someone,
How's it going?
anyone, working on a cure. Over 3 years, the Ryan Foundation graduated from bake sales to $100,000 golf tournaments with Ryan as their poster child.
Show you this? Yeah, it kind of shows Ryan's love for baseball. Every time I walked into a corporation or a company or a store, I would show them a picture of Ryan. I would say, I'm here not as a police officer, I'm here as a dad, and here's my cause. And I'd show them the picture. If I went to 100 places, 99 would still say no. But one would say yes. And then that was a big day.
But his biggest day came in 1995 when Mark met a young scientist at UCLA named Emil Kakkis. Dr. Kakkis was one of the few researchers in the world studying MPS 1, and his meeting with Mark Dant couldn't have come at a better time.
You're almost ready to shut down?
The research was stalled, and I still held out hope that we might be able to find some angel investor to come in and give us money and make it happen, but it was very frustrating to feel that you had something that would be beneficial for a disease that wasn't currently treated and couldn't do anything about it.
So this is the lab where you actually do the research on the enzyme? Dr. Kakkis had a sense that this complicated disease had a fairly simple solution. Create a synthetic version of the enzyme that's missing in kids who have MPS 1. This is what a normal, healthy human body does naturally?
Yes, normally your body makes these enzymes all by itself. They're helping everything run because your body is constantly building itself and breaking itself down. And one of those enzymes that's involved in that process is missing in MPS 1 kids. So by making it, we can help substitute for what they can't do themselves.
The science was solid, but to take his synthetic enzyme from the lab to the bedside, Dr. Kakkis needed money. And Mark Dant promised to provide it because he knew that time was running out for his son, who by the age of 7 found even the simple act of breathing unbelievably difficult.
I'm getting paged again.
As Ryan fought for his life, Mark fought for money, eventually raising a total of $1 million. He gave all of it to Emil Kakkis.
They basically decided to take all their money, put all their chips on one number, which was us, and hope that their number would come up, and fortunately it did.
Dr. Kakkis attracted additional funding from a small biotech firm and had gotten FDA approval for a clinical trial of his synthetic enzyme treatment. 10 kids were chosen for the trial at Harbor UCLA Hospital in California. Ryan Dant was one of them.
Well, guys, I think it's time to head towards the gate.
In February of 1998, Ryan's neighbors gathered at Dallas-Fort Worth Airport for a big send-off.
When I was at the airport, when I was about to get on the plane, everybody made arch, and I went through it and went onto the plane.
All the years of knocking on doors and holding bake sales had paid off, and as they boarded the plane, the Dants finally had something to hope for. At the hospital, there remained the reality of a sick little boy
I need a wheelchair, Mom.
who spoke at the time about what he was getting ready to go through.
I'm a little bit scared.
He was tired of needles and doctors and feeling weaker and weaker every day.
When's this gonna end? I just want to go home because I don't like it here. And I don't like to do all these tests.
What's going to happen, that enzyme's going to circulate in your blood, in your body, going all over your body, and get to where it's needed to start breaking down the material that's there.
2 months before Ryan's 10th birthday, Dr. Kakis and the dance administered the first dose of Ryan's enzyme treatment.
Okay, ready? I'll bet $10 that 1 week you're going to feel better. No?
Ryan wouldn't take the bet, but it was a bet Dr. Kakis won. And he wasn't the only winner. Week by week, drip by drip, Ryan's life improved.
Here we go. 1, 2.
Yay! And here he is today, at age 13, back in the game, a day his parents thought they might never see. Hey, that was a pretty good hit.
Thanks.
Before you started getting your treatment, could you hit like that?
No,
What were you like before?
I couldn't hit the ball a lot. I couldn't actually run that fast.
So you didn't have much energy.
I couldn't bend my arms back.
And you used to have headaches, your father told me.
Terrible.
How bad were they?
Really bad. As bad as you can think.
But the emotional pain often was worse.
I was just getting scared coming up to that 10 years.
So you actually knew there was a possibility before that you might die when you were 10?
Yeah.
And you thought about that? You worried about it? What'd you say to your parents?
It's gonna be okay.
And now that you're on the treatment, what's happened to all those symptoms?
Feels like I can do everything that I ever wanted to. Like go ride my bike every day or play baseball, basketball, you name it, I can do it. Except rock climbing.
Which you probably shouldn't do anyway,
Yeah.
right?
And Ryan wasn't the only one from the FDA trial to get better. All 10 kids showed improvement.
Time out.
Results so impressive they were published in the New England Journal of Medicine. 3 years after that first trial, Ryan still receives the enzyme treatment once a week at a Dallas hospital. And with him is this boy, Spencer Holland, whose own family is facing a crisis similar to the dance, only 3 times worse.
On the counter.
Like a puppet or something?
Amy and Steve Holland have 3 kids, and all 3 suffer from MPS 1. When the clinical trial started, the Hollands were told only one of their children could get the drug. They were asked to choose which one.
How do you make that decision?
How could any parent make that decision for their child?
So they left the decision to the doctors, who chose the oldest, Spencer. Like Ryan, he has improved tremendously, but for the Hollands, it's been a mixed blessing.
We saw Spencer getting better and stronger and more like a normal, healthy child. Meanwhile, our little girls were getting sicker and weakening. It seemed like every minute that ticked by, we knew that there was a drug available to help our children, yet we couldn't get it to them.
At that point, the drug still had not been approved for general use, and it still hasn't. The FDA is requiring a second clinical trial with a larger sample, one with 42 kids worldwide.
What did you think when that happened? That must have been bittersweet for you.
When we heard about the second clinical trial, of course we submitted both of our girls' names, but we never thought that both of our girls would be chosen to participate. So every day we waited for news to hear whether or not they would be chosen.
Both girls were chosen, and just in time. Lainey and Maddie Holland had been deteriorating quickly. Lainey had to have brain surgery, and Maddie was losing her ability to walk.
Ready to load up?
Recently, the entire Holland family had to pull up roots and move from Texas to North Carolina for the experimental treatment.
At this point, we would be willing to fly to the moon and back once a week, I think.
Just to get
just
this
to get
treatment.
this drug.
Although the Dants and the Hollands consider this treatment nothing less than a miracle, it is a miracle with limits. It hasn't reversed all of the damage in Ryan's hands, and doctors suspect the enzyme is not reaching the brain. That means it probably will not stop the mental deterioration typical of MPS 1 victims. But thanks to the work of one visionary doctor and one determined family, these children now have something precious: time. Time to await a cure and plan for a future that many thought would never come.
If you ask Ryan when he was little what he was going to be when he grows up, he would always say a baseball player. And for a while there, he stopped saying that. Now he says it again. He still believes he's going to be a baseball player.
He's got dreams again.
Right. And so do we. I think Ryan has the same potential that he had when he was laying as an infant in the nursery. He has the potential to be a great person.
Back when he was perfect.
Yes. And he still is.
So I've seen that video so many times, but I do love sharing it over and over again because it highlights many aspects of the disease, the impact on the family, the kids that are in the trials. We do have kids that come from all over the country. Some move to the United States from other countries just to be in these clinical trials. And so that impact and that burden that's placed on the families is really important, I think, for us to recognize and appreciate. But on the brighter note, this did ultimately result in the approval of Aldurazyme back in 2003. So going from that small bungalow to a bake sale that Mark Dant and his friends started, to the spinoff of BioMarin. So BioMarin was the company that was that industry sponsored. The funding that he mentioned, Emil Kakis mentioned, the collaboration with a pharmaceutical company, that was BioMarin, and they ultimately got it approved in 2003. And this is Ryan graduating from college. And since 2003, since this initial demonstration of the efficacy of enzyme replacement therapy for lysosomal storage diseases, there have been 4 additional enzymes approved for 4 different MPS disorders and multiple other enzyme replacement therapies for other lysosomal disorders such as Gaucher disease, Pompe, and Fabry disease. And so tens of thousands plus more patients, families really impacted, lives saved from this initial discovery of cross-correction of enzyme. But as they mentioned at the end of that video, enzyme is not a cure for any of the MPS disorders. Unfortunately, what we have found both in our clinical care as well as natural history studies, some of which I have done, for example, in the bone and joint disease over time, enzyme does not get into the bones or joints well, and it also cannot cross the blood-brain barrier. And since it can't cross the blood-brain barrier, it really is not thought to have any effect on CNS disease, which is an important and critical problem in multiple MPSs. And there are a variety of solutions that have been looked at to address this one problem of getting enzyme across the blood-brain barrier. And this is the area I'm going to focus on today. I'm not going to talk as much about the bone and joint disease as we're really going to focus in on how do we get enzyme into the brain to address this severe neurodegeneration, behavioral problems that patients with neuronopathic MPS have. And the 4 approaches that I'm going to go over today are 4 approaches that have been developed and pioneered really in the MPS disorders, both here on campus as well as in the UCLA campus. And these are intrathecal ERT, antibody ERT fusion protein, neuronal progenitor cells, and gene therapy modified hematopoietic stem cell transplant. So intrathecal enzyme replacement therapy. This approach to drug delivery is used in a variety of diseases. It's very commonly used in oncology as well as pain management, but it's being applied in MPS because it's a way, as I said, to get that enzyme to the brain. And the way this is done is by either taking a needle, as shown here on the left, and inserting it into the intrathecal space, or more commonly now, there's a catheter that's implanted. The catheter goes into the intrathecal space, and there's a port that's implanted under the skin where the enzyme is given and it gets into the intrathecal space that way. And during her time here at Lundquist, Dr. Patty Dixon really spearheaded this initiative in MPS. She was the first person, along with the collaboration with Dr. Emil Kakis, to show that intrathecal ERT improved functional and biochemical outcomes in an MPS 1 canine model. And then they took this on to start the first clinical trial using intrathecal enzyme replacement therapy in any of the MPS disorders. And following on that first phase 1 trial, Dr. Agnes Chen, who is an investigator here at Lundquist and the division chief of pediatric neurology at Harbor, once Patty left, Agnes has continued this program on and has done the long-term extension studies for intrathecal therapy in MPS 1. In addition, this work then, seeing this already done in MPS 1, was further expanded by pharmaceutical companies to address the neuronopathic disease in MPS 2 as well as MPS 3A. Now, sadly, these trials did not reach their primary outcome for efficacy and were closed early. Because if this had all worked, then I guess my talk would probably stop right now. But sadly, there's had to be multiple other approaches looked at for treating the CNS disease. But this was really the first idea of how to get enzyme into the CNS. The next approach that really made a large impact on the field was developed by Dr. Bill Partridge at UCLA. And this approach is shown here in the schematic. It relies on the ability of the transferrin receptor to shuttle iron from the vasculature into the brain. And so understanding this shuttling of iron, Dr. Partridge applied this to getting enzyme to the brain. And what he developed was this transferrin receptor targeting antibody that he fused to the enzyme. In this case, it was iduronate 2-sulfatase, which is the enzyme missing in MPS 2. And by doing that, the transferrin receptor, this fusion protein will bind to the transferrin receptor, and then the receptor shuttles it across into the brain and gets it across that blood-brain barrier. And this initial preclinical work by Dr. Partridge was then carried on into clinical trials, initially by JCR Pharmaceuticals, which is a Japanese-based company. And they were able to show safety and efficacy and get the enzyme approved in Japan in 2021. They're now doing their trials in the United States to move forward and hopefully obtain approval in the United States as well. Another company, Denali, has taken a similar approach with a similar transferrin receptor antibody and has looked at this in MPS 2 as well. They are currently under review at the FDA, and I think their PDUFA date is in April, so some exciting news hopefully coming out there. And both companies have now moved on to looking at MPS 3, which is one of the other very severe neuronopathic forms of MPS. And so this is a very exciting area that shows a lot of promise, but we'll have to wait and see long term what the effects will be. Now, looking again back locally here, another really exciting approach that's being developed by our colleague here, Michelina Iakovina, is using neuronal progenitor cells. And she's looking at this for Sanfilippo type B. And how this works is she develops these neuronal progenitor cells, and they are cells that will produce the missing enzyme. So for Sanfilippo B, this would be NAGLU. And those cells are then injected directly into the brain. And when she does that, the cells can then expand out from that location. And the idea and the hypothesis of how they would fix the CNS disease is that they would have that same cross-correction that I already described. So the new cells that are secreting NAGLU, they would secrete that out. Cells nearby that don't have the enzyme take that up and get fixed that way. But the other really interesting part of this approach is that because these are progenitor cells, they are thought to be able to then mediate regeneration. And the hope is that that would end up with restoration of some of the neural network. And this is a program that Dr. Michelina has funding from the California Institute of Regenerative Medicine to work on. Now, the last approach for what I call enzyme restoration, so all of these approaches are focused on getting the enzyme back into the cells. And this last approach was actually, it was not developed here, but Dr. Cohn up at UCLA has done a lot of the work in this for other diseases. So this approach has been used for it's approved for sickle cell disease. It's also approved for a few inherited metabolic disorders like metachromatic leukodystrophy and adrenal leukodystrophy. But it's now being applied to MPS. And part of why it's being applied in MPS was that many years ago, in about 1985, I believe, the first transplant, so an allogenic transplant, so more of the standard transplant we think of another individual's stem cells being transplanted into an individual with the disease. This was done for the most severe form of MPS I called Hurler syndrome. At my last institution, University of Minnesota, where they've got a very large transplant program now for Hurler syndrome. And it was shown to be effective. And so it has been the standard of care for Hurler syndrome, the most severe form of MPS I, for the last 30, 40 years. Unfortunately, when this has been tried in the neuronopathic forms of MPS II and III, it doesn't show the same efficacy. And it's not quite clear why. And so the approach that's now being taken is this autologous hematopoietic stem cell transplant with modified stem cells that are modified in a way to increase the secretion of enzyme. So basically what is shown here is that you mobilize the CD34 stem cells, and those are taken into a centralized facility where they have the gene that is missing for the enzyme placed in them with a promoter that pumps out tons of enzyme. And so in this way, not only are you getting functional cells with functional enzyme, you're getting cells that make a lot of enzyme. And we do think that the higher dose of enzyme does seem to improve some of the outcomes, particularly in the bone and joint disease, possibly the heart disease. On top of it, because you've got microglia in your brain, which are hematopoietic in origin, these cells will migrate into the brain and be able to secrete enzyme in the CNS and hopefully correct disease there as well. So it's an exciting new area, kind of pulling together all of these different aspects of what we know about how enzyme should work, how it will be most effective. And the other benefit to this is that because it's autologous, you have much lower risk of complications. You don't need as much PrEP chemotherapy, and you don't have that risk of acute or chronic graft versus host disease. And so the adverse events, the side effects, the problems with this therapy are much better than with the allogenic hematopoietic stem cell. And here too, again, I think these videos can say so much. I wanted to stop. This is a shorter video, but this is about a patient of mine and Dr. Chen's, who we see at Chalk Children's Hospital in our MPS clinic. And his name's Ollie. He has neuronopathic MPS II, and he has been the first boy ever treated with this approach.
A 3-year-old boy from California with a rare debilitating condition has become the first person in the world to be treated with a gene therapy developed in Manchester. Ollie Chu has Hunter syndrome, which causes progressive damage throughout the body. Our medical editor, Fergus Walsh, has his story.
This is a story of hope.
The future for Ollie seems very bright, and hopefully this means more kids will get the treatment, and the future will be bright for them.
How science is taking on a brutal inherited disease.
We're so happy, so pleased with his progress. His results are amazing.
And what the new treatment means to those affected.
It's really amazing how this treatment has achieved, and how he is so different now than before.
Oliver, just check your temperature. Thank you, that's a good boy.
Oliver has Hunter syndrome, also called MPS II. A faulty gene means he can't produce a crucial enzyme. This means chemicals build up in cells, causing progressive damage throughout the body. Usually only boys are affected by this rare inherited condition.
Boys, when they're born, usually look really well and healthy, but gradually over time those chemicals build up and they gradually cause each part of our body, our bones, joints, brain, or airways to become more and more diseased. Usually wheelchairs are required by the end of the first decade. The average survival was in the teenage years.
It's December 2024. Oliver has flown with his dad from California to Royal Manchester Children's Hospital for the start of a groundbreaking medical trial.
And everything's a race against time for us. You know, at the end of the day, we just want the kids to be, you know, healthy and have a more normal lifestyle than they have right now.
Oliver is having his blood filtered to remove stem cells. These special cells are going to be tweaked, genetically altered in the lab.
So these are Oliver's stem cells. It's taken just over 3 hours to collect them, but this is just the first stage.
Oliver's cells are transferred to labs at London's Great Ormond Street Hospital. Scientists use a harmless virus to insert a working copy of the faulty gene into each of Ollie's stem cells. The gene gives instructions to the cells to start producing the missing enzyme, which should get to all parts of the body, including the brain, with the aim of halting further physical and neurological damage. It's February, and Oliver is back in Manchester, this time with his mum.
We have to get bag one first.
His genetically altered stem cells are here too. Deep frozen, the cells are slowly brought to body temperature.
Hello. Starting now.
And this is the big moment. The infusion of the gene therapy. Oliver is the first child in the world to be given this one-off treatment to combat Hunter syndrome.
That's us done.
Researchers at the University of Manchester and clinicians have spent 15 years developing the gene therapy.
So having an effective treatment that deals with all aspects of Hunter syndrome would be utterly life-transforming for families. It would mean moving from a constant life of medical interventions and slow decline and an early death to a life where there's hope.
Ollie, you want to go play?
3 months later, the entire family is over from California, including Oliver's brother, Skylar, who's 5. He also has Hunter syndrome, but was too old for this trial.
1, 2, 3.
Skylar receives a weekly infusion of enzymes. This helps prevent damage to the body, but they don't get to the brain. By contrast, Oliver has been able to come off all treatment because the gene therapy is working.
I want to pinch myself every time I tell people, you know, Oliver is making his own enzymes. Every time we talk about it, I wanted to cry because it's just so amazing.
My wish upon the star is for, you know, Skylar to be able to get the same treatment. As devastating as the news was that both kids had MPS 2 Hunter syndrome, with this gene therapy, it feels like Oliver got a reset in his life and gets a start, you know, with almost a fresh slate, you know, and that's why I want the same thing for Skylar.
The trial is funded by a medical charity, LifeArc. Unusually, there's no pharmaceutical company involved. 9 months on, doctors remain cautious but are thrilled. The ambition is it could be a permanent fix for Hunter syndrome.
We're so happy, so pleased with his progress. His results are amazing. I've been waiting 20 years for this to see a boy like Ollie doing as well as he has, and it's just so exciting.
His speech has gotten exponentially better, his mobility has gotten better, his agility, his cognitive, everything from his learning curve hasn't just gone up, but it's like shot up for that. So like no more medication, no more infusions, like as close to a normal life as we can give him at this point.
That's extraordinary.
Yeah, that's all we dream for, you know, for both our kids, honestly.
Ollie is one of 5 boys who are part of this world-first gene therapy trial. It's early days, but if successful, it would transform their life chances and give hope to other families affected by this rare and often devastating condition. Gene therapy is making huge strides in tackling rare diseases, so the outlook has never been so promising. Fergus.
So, it's pretty exciting to see a boy like Ollie get a treatment like this and how well he's doing. And I can tell you, seeing him in clinic, it's really, really amazing to see. Sadly, though, as this dad and the mom pointed out, he has an older brother, Skylar. And if you recall in the first video, that second family had 2 older siblings also who had MPS 1 and who didn't qualify for the study initially, and who luckily later were able to be in a trial. But what we find is for these therapies that are directed at the CNS disease, they're really not effective unless they are started very early in the disease course. And so just like Alzheimer's disease, this loss of intellectual function, the loss of the CNS, the cells in the CNS, the cognitive function, the behavioral things, once they start to get all these different symptoms, this is not reversible in terms of the CNS disease. And this is most relevant now for MPS 2 and 3, because as I mentioned, the severe neuropathic form of MPS 1 is really treated with transplant now. But this is still a large part of the MPS population, and we estimate really greater than 99% of the MPS 2 and 3 patients do not qualify for any of these therapies. Even once FDA approved, they're unlikely to help them with the CNS disease. And so their families are stuck with a variety of other symptoms, challenging behavioral symptoms, things like chronic diarrhea, sleep problems, that really impact the quality of life for both the patients and the families. And so this is where my work has been focused on the last 10 years or so, because there's been a lot of discoveries at the same time that people are figuring out these new approaches to therapy. There's been basic science work done to really understand what are all of the impacts of this loss of enzyme. And so we focused on the fact that there's this deficiency of a lysosomal enzyme and that we want to give back that enzyme. But beyond having that deficiency in the lysosomal enzyme, the problem is when you have that, and then you have the GAG storage in the lysosomes become dysfunctional, and that causes all sorts of issues with impaired autophagy. You get mitochondrial dysfunction. Those lysosomes that are chock full of GAG, that gets released into the extracellular matrix, and now we understand that that goes on and can trigger inflammation throughout the body. And that inflammation then is contributing to a lot of the disease symptoms that we see in these patients with more advanced disease. And so this is the area that I've been focusing on with repurposing medications for the MPS disorders. And today I'll finish the talk by describing a clinical trial we finished a few years ago here at Lundquist that was a phase 1/2 clinical trial of anakinra in Sanfilippo syndrome. Now this work, I couldn't talk about the trial without acknowledging the really important work done by Dr. Brian Bigger and his group at Manchester. Who really defined the pathway and the exact mechanism in MPS 3 for the role of inflammation in disease. And the study that I show here is a study where they looked at a wild type mouse, an MPS 3A mouse, the MPS 3A where the IL-1 receptor was knocked out, and then a wild type mouse with IL-1 knocked out, and then let me rephrase that. They looked at the 3A mouse, the 3A mouse with the IL-1 receptor knocked out, and then they had one where they did a gene-modified transplant where they gave back IL-1 receptor antagonist to be overexpressed in that mouse. And what we see is that when they have this overexpression of IL-1 receptor antagonist, you have decreases in your astrocyte and microglial activation, shown here in the bottom left, and improvements in memory and decreased hyperactivity. And so both physical improvements as well as behavioral cognitive improvements in the mouse model. And with this information, we were able to go to the FDA and get an IND to try this in children with Sanfilippo syndrome. And so why did we pick anakinra? Well, anakinra is a recombinant interleukin-1 receptor antagonist that crosses the blood-brain barrier. So some of the treatments, the anti-inflammatory treatments, don't cross. Most of them actually don't cross because they're large antibodies. But since anakinra is a specific antagonist, it's small and it can get across the blood-brain barrier. In addition, it mimics the model, the mouse model that Dr. Bigger had done, where he was overexpressing IL-1 receptor antagonist. And so again, we obtained approval by the FDA to treat Sanfilippo syndrome, in part also because anakinra is approved by the FDA to treat other pediatric neuroinflammatory disorders. And this trial was an open label clinical trial. We had 8 weeks of observation in the beginning, 36 weeks of treatment, followed by 8 weeks of observation at the end. And I'm going to really mainly focus on this outcome. This was an outcome that we designed specifically for this study because of the heterogeneity of MPS III. So what we understand from the past clinical trials is that not all kids have the same set of symptoms. And so if you choose one outcome, there might be some kids in your study who actually don't have much problem with that in that one area. And so we asked caregivers to choose their top 5 prioritized problems from this list that came from a previous caregiver focus group. And then they rated those 5 problems at each visit in terms of how stressful, how impactful they were to their lives. And from looking at it this way, we saw that during the observation period, the initial observation period, it's relatively flat. And then with treatment, we had an improvement in this individual clinical response. And when we stop treatment, we see this worsening or this rebound off treatment after 8 weeks. Another approach that we use for addressing this heterogeneity of disease is called the multi-domain responder index. And so this is a way that, again, looks at a variety of different aspects of the disease. And so each of these outcomes here on the left are an individual survey that the parents or caregivers filled out. And so we look at a minimal clinical improvement, so we can define a minimum amount that would be considered clinically significant, and we define whether they've improved, no change, or worsened. And so what you see here at both 8, 16, and 36 weeks, on each of these outcomes, we have a significant number of patients improving on anakinra, and a fair number, surprisingly, just with no change. When they came off treatment for almost every single outcome, we saw a similar pattern to what we saw on the slide before, where things worsened on those 8 weeks off anakinra. And finally, we wanted to look at some of the biochemical effect of anakinra to have a biochemical confirmation of this clinical outcome that we were looking at. And so what this shows you here is, again, taking this multi-domain responder index on the left and looking at that in relationship to the changes in the immunophenotype. And so we would expect with an anti-inflammatory approach that you would have an increase in CD4 T cells, these are your helper cells, your Treg cells, a decrease in your CD8, which are more of your cytotoxic T cells, a decrease in your monocytes, and really no effect is thought to, anakinra is thought to have no effect on B cells. And this is what we saw in most of the participants. And the other interesting thing that we noticed was that the ones that had the greater increase in T cells had a greater number of outcomes on this MDRI that had improved. And the ones with the greater decrease in their cytotoxic T cells, again, had the greater number of improvements on the MDRI. So giving us a biochemical validation of some of the clinical outcomes we had noticed. So to summarize, I've gone through the early biology of the discovery of cross-correction by Dr. Elizabeth Neufeld and their preclinical work that led to the first ERT study in humans with MPS. We then talked about the early work by Dr. Dixon, Dr. Chen for intrathecal ERT, and that expansion, this rapid expansion of ERT therapies for the MPS disorders. And following that, there's been this rapid explosion of different approaches to try to get that enzyme into the CNS. And I think we're now at a point where we have one approved in Japan and 2 very close to approval, likely here in the US. And there's just a lot of other exciting approaches that are being developed. And so I think there's a lot of hope going forward for the CNS treatment in MPS. And finally, I've spent the whole talk acknowledging investigators and the work they've done. I wanted to just finish with this slide that all these are patients that have been in my clinical trials and natural history studies over the years, and just to acknowledge really the commitment they have in making these trials work, in moving the research forward. MPS has been an area where we have never worried about families participating, the patients wanting to participate. They have been strong advocates for research funding, and I just wanted to acknowledge them at the end here. So with that, thank you very much for listening, and I'd be happy to take questions. Yes. You're welcome.
Beautiful. It's blinking. Sorry about that. Yeah. And so I have two questions, but I'll have the first one first. With your San Filippo study with anakinra, there were patients who didn't respond, it seemed. Do you have any idea why? Do you have a hypothesis as to why some did well and some didn't?
Yeah. We did not, we've looked into different characteristics, different baseline characteristics of who responded and who didn't, and have not found anything distinct of the non-responders. And so I think in terms of why, probably because those patients, the inflammation is not as an important component of their disease symptoms. As I showed in that slide, there's issues with autophagy, there's mitochondrial dysfunction. It's such a complicated, multifactorial disease at this point that my guess is that it's just less inflammatory in some patients than others. The other thing that we really saw even more so was some patients just couldn't tolerate the daily injections. And so some of the people that were non-responders, it might have simply been that they just weren't taking the medication, they weren't able to do the injections, because to take this medication, it's a daily subcutaneous injection, and the dose needed is actually 2 injections. So the families are giving 2 subcutaneous doses, like an insulin injection, each day. And to do that can be quite difficult in a child with aggressive behavioral issues and so forth. So I think that maybe there's some different underlying physiology related to the role of inflammation, but there's also the fact of, did they actually receive the medication or not?
If I may, a very different kind of question.
Okay.
So, there were some limitations with enzyme replacement therapy. It really can only treat very specific kind of disease.
Yeah.
But as these things have developed and you have these newer attempts and approaches, do you see, like, for example, some of the approaches you're using now as being useful to other kinds of diseases that more broadly, perhaps other rare diseases? Is this something that we're going to learn some general basic principles for disease treatment from that we can apply to other things?
Yes, absolutely. So a lot of the approaches that I discussed today are being used in other lysosomal disorders. The anti-inflammatory approach has been used in not just other lysosomal disorders. So Gaucher is another one that it's being looked at in, but there's a whole variety of neuroinflammatory conditions that that's being looked at. And then the approach to getting enzyme across using the fusion protein approach, that for sure is being applied to other CNS diseases to get medication across. So what we do in rare disease, I think this is your point, can be applied to more common diseases, for example, in oncology.
You said you had to start the treatment when they were young, but they don't show any signs at birth. So how do you know that there's an issue early enough to be able to start treatment that's then going to be helpful?
Yeah, you bring up one of our biggest problems in this area right now. And as you saw, it was the younger sibling in that last video I showed. So he was picked up because his brother was diagnosed. And so they found him, he diagnosed him younger. Newborn screening for all of these disorders would be amazing and a game changer. We don't have all of them on the newborn screen for a variety of reasons. The main one is that the standard has been that there needs to be a treatment available to have something added to the newborn screen. The catch-22 there is that you can't get your clinical trials done because you can't pick up the patients because they're not in the newborn screen. And so what happens is it's often the younger siblings of the older siblings who got picked up clinically that end up in these trials because of that reason. And that's also why in that slide I showed you, it's probably greater than 99% of our patients don't qualify for these trials. So hopefully in the future, we can get all of the lysosomal storage disorders on the newborn screen in a way that would help us really move the trial enrollment and the therapies forward and be most effective.
And there's currently no ERT for syndromes 3 and 5?
There's not. No. ERT has been tried because Sanfilippo is primarily a CNS disorder. A enzyme that only impacts the peripheral, the somatic disease, has not been what's been sought after. And so there have been enzymes tried, but since they weren't successful in the CNS disease, they did not go through to approval. Okay. Well, thank you, everybody.